Okay, here it is. Okay, maybe go ahead and dive in. Just today's giving you just updates, papers, things we've missed over the last few weeks. Happy New Year, everyone, and hope everyone had a good holiday break. Um, one thing is, I'm gonna switch this to bi-weekly meetings, so every 2 weeks. After today, you get a calendar update. I'm probably gonna switch it to every 2 weeks, just for saving people's time's sake. That'd be probably better. And if people know of speakers and people want to give presentations at these meetings, let me know. Always welcome to have people talk at these meetings, and you can hear less of me, which would be probably good. All right, so the news, yeah, just papers. Hopefully this year we're gonna have some more papers coming out. We're working on a few right now, so probably expect some later this year. Trying to still do this in-person meeting in Malta. We'll just have to waiting for some potential sponsors, and well, maybe a month or two. If you don't hear anything, then probably have to shelve it. But if people know of sponsors that might want to help, let me know. Okay, so I'm gonna first start by going over what the dominant variants are, and then what the cases are, and as probably people have heard. The most dominant variant is this nice distribution from Raj's platform dashboard. Is JN.1 is basically everywhere. That's the one that has all the mutations that we'll go over. And the wastewater is showing indeed, as people probably heard about and know about, the increase of wastewater is now the second probably biggest wave since this big peak of Omicron that happened back January 2022, 2 years ago. So that's not a good thing. And then you see regionally, kind of the Northeast is spiking more than other regions, but everywhere it's trending up. This is Middlesex County where I am at, but most places seem to have these kind of trends. And this is updated at January 9th. So this is from after the holidays. So some people are predicting the models that this is going to start tapering off soon and plateauing, but we'll see what happens. The CDC has a nice wastewater site that they had updated a couple of months ago, I think now. But there's I like to show there's two of them. It shows the same thing, but but you can see regions they claim that have the highest. They're saying the Midwest probably has the most highest active wastewater for COVID-19. You can see the distribution in states. Some states don't have enough sufficient wastewater data to report, but states that do, you can see most places are very high to very high. Some places are moderate, but JN.1, as we talked about, is the most dominant variant now in the US at least, but in the world it's the same. I'll show some of that data coming up. And then, yeah, as I said, agreeing with the wastewater from the sequence data, the CDC is, you can see JN.1 how quickly since before the holidays till now. This is updated from 6 days ago. 1/6 is the last estimate they have from the nowcast estimates. You can see JN.1 is pretty much becoming dominant more and more. And just in case you want to know, to remind you which lineage it comes from, is from that BA2.86 lineage. And then globally, as we would expect and we've heard, and I'll go over some of the news now, the hospital admissions for the countries that do, or regions that do report it, like the US, we see that this is going up pretty drastically. It's a lot lower than obviously the first Omicron wave and our initial waves, so that's a good sign, but it's still going up. All right, so let's dive into the news now that you get the update on how the virus is going. So if you hadn't ordered your free tests back in November, they had the White House in the US, the administration said they will offer more for free tests. But you can still do it. Go to this site here, covid.gov, order your free home test if you haven't already. And it is showed you from the spike and everything that's happening, as you would expect. You got still 1,500 Americans, or roughly that dying. And in the world is pretty much, agreeable with these numbers for the most other countries. Every week, this article here talks about why is that case. Well, as you would probably not surprise anyone here, is because of this low booster vaccination rate that's occurring in children and adults, which potentially these deaths could be reduced, right, if people have given their boosters and especially the newest booster that would cover the new variants. This nice Time article was talking about, is it dangerous to keep getting COVID? As we know, a lot of people keep getting reinfected, and it's probably no surprise to anyone here. But there's some nice quotes from Ziad Ali and Davey Smith talking about, yes, every time you get SARS-CoV-2, infected with SARS-CoV-2, this could be adding more and more impact to your immune and system, and reducing your ability to actually reduce the chances of long COVID and reduce the chances of having long-term impact 6 months or down the stream, as most people here probably not surprised. And I said again, the variant that's JN.1 is just another article talking about the UK, talking everywhere in the world is JN.1. That's been called the Juno, I guess, where it's been becoming dominant. In the UK, they're saying that probably, as the predictions go, this rise is probably getting to a point, which, the good news in quotes is that it's getting predicted to a point where it's reaching that plateau, which people are thinking it is. And as most typical variants in the past, you see that plateau and go down until there's a new variant that happens. But in the meantime, the WHO has reported, from the 1,500 deaths in the U.S., but total, like they say, from last month is 10,000 that have died. Although it's low in the entire populations of the world, it's still 10,000 deaths that could probably be reduced if people get their vaccines.
After I didn't understand what you said about multiple COVID, the point you made. I mean, I didn't quite get what the point you made.
Oh, the previous one? Yeah, so the previous one say, when you get multiple infections, everybody will show you some papers on this too. So the more and more infections you get, some of the work that Ziad Ali has done and others have done, they show that it will keep the inflammatory conditions that are created every time, and the immune impact that's created every time, and then we're talking about mitochondria, it compounds. So every time you get the infection, it's going to add more stress to your body. And I'm oversimplifying it, right? Stress to your body to the point where your immune system will become less and less able to behave how it should, for people who would develop long COVID. So that's sort of the idea in these papers. It's saying, getting repeated infections could downstream have pretty detrimental effects for some people, not all.
And is that true even when people feel like they've recovered from each of those successive infections? Because I certainly know people who are on their, second or third infection, and they feel, like they've recovered, so they're not really concerned. They don't seem to be particularly concerned about it.
I mean, it's from the papers we've seen, and we talked about it in the past. Even people who recover, this seems to be, I don't know the percentages, but we've seen cases, and reports and papers to say that people who recovered do have long-term effects, but they just don't know about it, until it's too late, until they do the studies and show, this organ's impacted this way and that. There's some papers, and I'll show you coming up, that start going over this stuff too. Even people who feel fine. I mean, it's not everyone, but we don't know the numbers, right? It's kind of the research area that needs to be done. Right. But it definitely, for some people who feel fine, I'll show you some of the long COVID papers that have come out in the past 2 weeks that would indicate even mild patients get some long-term effects they might not even be aware of until later. But so that's the case where and I'll show you some data and papers too, talking about how the vaccination will greatly reduce the chances, as we all know here. So that's how they get promoting vaccines. And this big wave, how big it is again, talking about the US, we already went over that, but it's as the wastewater show, it's pretty very high viral activity happening there, and people should be concerned. So then Eric Toppel's blog back 2 weeks ago, because we haven't met since the New Year, or a week ago, January 5th, he did a nice little synopsis of what's the state of the current pandemic. As I said, in the second biggest wave, JN.1. Here's a nice little plot. It wasn't on this blog post, but it was on his Twitter page, where you're showing, again, comparisons to previous waves. You can see the first Omicron wave is off the chart here, but then JN.1 is from the wastewater levels. You can see how JN.1 is pretty much, it's still going up, but people are hypothesizing that it's going to plateau soon. Based on,
I'll shoot the question at the last statement. What are the parameters that allow them to hypothesize? that it'll hit that inflection point peak.
There-
I
yeah,
just, I wasn't sure how you make that prediction. I was just interested.
yeah. I mean, from what I've read, it's basically the models they have is based on previous strain behavior. So if this behaves like previous strain behaviors, their models would be saying in the next couple of weeks it should behave this way. But if it doesn't, their models could be wrong, right?
So it's like that wave of infections that you
Exactly.
once initially pointed out seems to be a curve that accompanies each one of these, I guess, right?
Exactly. So they're kind of in their models, from what I understand, they're accommodating for, let's say, the amount of population that gets infected and the peak of the when that would happen based on that region. And then they'll say, well, that's the peak. You know, there's not going to be that many more infections after, you know, let's say I'm making this number up. 60% of the population is infected. You know, so that number's made up. But that's how they kind of assume. Like they look at the previous waves, and then they look at how many people in this case, based on the wastewater, are being infected, and then they model that with their models there. If that makes sense.
Oh,
But
it
there's,
does. And thank you. It does make sense. Yes, thanks.
but you know, as any model, you know, as Eric Topple points out in here, you can see the number of mutations JN.1 has compared to the original XBB.15, and XBB.15 had plenty of more mutations originally than the previous strains. So JN.1 could have enough mutations to make it, the model is not as accurate. But we'll see what happens, I guess, in the next couple of weeks. Hopefully it plateaus. But JN.1, due to their lot of mutations, does have this evasive abilities based on that. But we'll see how well it does. This is the worldwide view of JN.1. You can see everywhere, pretty much in the world, that does monitor and does the sequencing of the virus. You can see JN.1's dominant, as Eric Topple points out here. And he talks about the boosters and talking about indeed, the XBB.15, the booster, latest, the bivalent booster mix that is out, that people have been getting. There's been studies, as he points out here, that shows that there's 60% protection against hospitalization, emergency room to the JN.1, which these studies have come out, which is great. But there's a low number of people getting this booster, so that's not the good thing. And then he talks about the long COVID from the studies, and he says, from some studies, from these 24 studies and other things that were done with vaccine protection, and I'll show you some of the studies too that came out in the past 2 weeks, there seems to indicate a 70% reduction of risk for long COVID based on the booster getting infected with these, the BA2.86 variants, meaning the JN.1. So those are the positive signs. If you did get your booster, you can get, yeah, go ahead, Henry.
Is there an explanation for why there's such large swaths of the world where it doesn't exist?
They don't have the sequence monitoring that other places do.
It's just a matter of monitoring.
Exactly.
Oh, okay.
Yeah. So these other places have been reporting the sequencing constantly, and these other regions have not. So yeah. So the white spots don't think it's JN.1's not there. It's just lack of monitoring for those countries. So there's now going to memory response and how well, talking about the vaccines and how well, let's say, previous infections you might have. These two papers came out in Cell Host Microbe just January 10th. So was that yesterday? 2 days ago? 2 days ago. And they're basically, both of these papers are talking about how there's a preserved T cell and I'll share all the slides like usual with you so you get all the links. But preserved T cell recognition response, even for like this paper, a hypermutated version of BA2.86, which would be the JN.1, I guess, equivalent, you could say. That's a hypermutated version. At the time, I guess, when they did these studies, obviously, JN.1 wasn't pretty dominant yet, because, as we know, papers are done months before the acceptance date. But what was nice about these papers, you could read about, but the abstracts and the little highlights here is that, for example, this paper is talking about the memory of the T cells, the response to SARS-CoV actually lasts a pretty long time for what they're finding. So, in the vaccines, we know there's like a 4 to 6 months waning period. Once you get your vaccines, you got about 6 months where it starts waning, your effectiveness of the vaccines. And it's probably good to get the booster, I would imagine, every 6 months. Here they're saying, well, with the T cell memory response, has a longer durability based on, the variants that are popping up now. So this study here was done in the South African population that were vaccinated and not vaccinated, if I remember correctly. I kind of skimmed through these papers, and this one was just a brief report done looking at different studies, with how well, as you can see from this population. Here they looked at a chronic lymphocytic leukemia population, and in a healthy condition. So,
Could I ask something?
Huh?
So when initially in the first 6 months, presumably you have this big antibody response that wanes, and
right,
then you have this memory response, but the antibody response doesn't seem to prevent people from actually getting COVID, because people get COVID whether or not they have high antibody titers.
right.
So do
This
they
prevents you
have
from
less?
getting it. Yeah, go ahead.
So do they have less severe COVID as a result of having the high titers, or is it all the memory? Is all the hard work being done by the memory response?
Well, yeah, I think the antibody is giving you the less severe case of COVID, right? It's going to reduce the symptoms and hospitalization, so it's less severe. But I think the T cell here, from what I'm gathering, and I could be wrong, is that the T cells might prevent you from actually getting, the infection to, enter your body. So I think that's what they're kind of saying. There's two responses, right? As you mentioned. So the memory of the T cells will probably be the classic immune response, from what I'm gathering. I could be wrong, but from what I'm gathering, that could maybe hopefully prevent the infection from spreading in your body. But again, this is the part of it.
I thought from what I read that the memory was actually what was leading to the shortening of the, like preventing the severity, even though the antibody response is not able to prevent it. But I don't.
No, the antibody response, from what I understand, is, I could be wrong again, but I think it's, maybe someone else could chime in too and to tell me if I'm wrong or not. Or I think the antibody response is going to help reduce the severity of how the virus will attack your body. If anyone else has any insight, you could chime in. But I think it's the more complex story that people are trying to figure out, right? What's happening with the T cells and other immune cells, and that's the part that's more and more papers are trying to come up with what you're talking about, Henry, trying to figure out what it's doing, right? So this leads to the long COVID now papers. So there's a nice NPR article about this paper here, and in one of our meetings with one of our groups here, I brought up this paper. Some of you have seen this. They're talking about this paper here looked at mitochondrial response. So this is good. This is in line with what we're working on too. So a bunch of people here, and they looked at the muscles, and they looked at they basically were quantifying different factors related with, like, for example, oxphos capacity, different factors of the O2 and respiration in the muscle, and they basically see that indeed for long COVID patients, I forget how long after COVID the long COVID patients were categorized. It might have been a year, but don't quote me on that. So I have to go back and read the paper more in depth. But this is from a population in the Netherlands. They saw that the classic thing some doctors say when you get to try to get over long COVID, start exercising. But as we know, a lot of long COVID patients with exercisers, it doesn't work with them. They have severe physical reactions. They're very fatigued. And so what this paper basically found is that, as we hypothesized too, is that the mitochondria is compromised in the muscle, at least, because they're focusing on that. So you can see there's some figures from here. For example, the red is the long COVID, the healthy is the clear. You can see for like oxphos capacity is being reduced. And this is this post, what does the E stand for? I forget what the E stand for, but this is the post-exercise, or malaise, I think. I forget what the PM stands for, but it's after, they did the one day after the exercise and how bad they felt after the exercise, because long COVID patients are not able to recover as well when they exercise. And this is their reaction. And you see the mitochondria is deficient. They're not able to go back to the healthy levels after the exercise. So this is and they did some metabolic studies too to find some key factors here.
And should they actually muscle biopsy these patients?
I don't remember what, I think it's
doesn't indicate that it's a circulating phenomenon that they
no, I think it's muscle biopsies they did from the Netherlands. The people from the Netherlands, yeah.
Not exactly non-invasive, but that's
it says that by taking biopsies is right here, right? Yep, biopsies, yep.
Wow.
Yeah, it's not invasive, right? It was a pretty invasive study, but it produced some nice, interesting results. Yeah, I was kind of surprised the participants would agree, but I guess at this point, they're trying to give everything for science, it sounds like, right? So, yeah, they didn't. I don't believe they looked at circulating anything in this, but I could be wrong. But from what they saw also was interesting, the tissue samples from the long COVID patient's muscle, muscle damage, there's immune response, you can see there, but they also saw buildup of microclots in the muscles. So that was interesting to read about. This other paper, I just emailed some people this morning because I just saw it, and it came out yesterday. So you might have not seen it, but this is a very interesting paper. I know some of the people here who have been helping analyze the Rob Schwartz long COVID data. The most recent thing we saw was that this adaptive immune response was pretty highly active in the long COVID patients, 8 months afterward. That was one of the striking things we've seen with that data. But then this paper comes out, so it completely agrees with what we find, which is good. But I think in our paper, we got to quickly try to get out because the field is moving rather quickly with this, and there's more and more patient samples and data coming up, which is good for the world. But what they found here is that indeed they're focusing on immune and inflammatory factors, and they're kind of focusing on what's happening for long COVID patients after that. So they compared, they had, the blue line here, for example, is the regular non-long COVID patients, and then the red line is this long COVID patient. And I forget how many they had in their study. I think it was like 40 or 50 patients they had. 43, here you go. The blood of 43 people with and without long COVID. So it's a smaller number, but they actually got some really interesting results. So one thing is that, you can they looked at some of the cytokines here, the CXR family, and you see that indeed in the long COVID patients, these things are pretty upregulated and active. They looked at other panels of immune markers and things related to the CD4, CD8 population of T cells, and indeed, which I'm not showing the figures here, but they indeed showed for long COVID patients, that there was a lot higher activity happening, dysfunction happening in these T cells. And one thing they said was an unusual high number of tissue homing of T cells, prone migraine in different parts of the body. And this is from the blood, so non-invasive.
So Austin, just quickly, in comparison to the data from Rob and the work that you've been doing, which incriminated in a big way adaptive immunity. Now they're saying T cell immunity. Do they equate? Are the changes as great for each? I mean, did they juxtapose and look at adaptive as well?
Yeah. So that's one thing they focused really on adaptive, as the title says, and they looked at all these markers. This is one of the plots here on the right. You can't probably read it right now on your screen, but it's heat map. They have some genes that overlap with what your adaptive, Steve, gene list was.
Right.
But so they have
it's not a B cell activity, actually.
No.
This is all focused on T cells.
Right. So they say that adaptive immune is really from the T cell dysfunction. That's what they're called.
Thank you.
Yeah. And I think they looked at CD4, CD8, I forget what else. And then they did a single cell RNA-seq, which they looked at a different cell population, like Rob's data. So that's a later figure. This is part of that here. This figure here, they looked at that, and they have some other figures related to that. But the good thing is all their data, they have single cell RNA-seq, bulk RNA, they have CyTOF data, even that it's all, the data accession numbers are there. And I think it's prime for us to jump on it and mine and analyze and compare to what we have, add to the pool of data we have. But yeah, this figure here is, I think this is figure 5 from the paper. You can see, for example, their little network of different immune markers that are up and down, like IL-6 and things like we know during the acute phase are really up. Those are not up anymore. Those are just no differences. And I'm not sure. I forget if we see that with our data, but we have to look. But the CCLs and other things that are really adaptive, you can see these are quite active and are up in the long COVID patients. Same here. You can see some of the HLAs and other things. They don't talk about mitochondria from what I saw, so I guess we're still in the running for that. But this is a good paper for maybe people wanted to help mine the data here. This could be something to push forward. Here's another long COVID data talking about the vaccine effectiveness, which I mentioned on Eric Toppel's blog. and to talk about there's two papers here that just came out in the past couple of weeks, some yesterday, some a few days ago. But for example, here is the talking about the vaccine effectiveness of preventing long COVID symptoms from UK, Spain, and Estonia. And they basically show here, this is one of the plots from the paper, but the basic gist of the paper was that they saw a substantial, like a 40% reduction in long COVID via vaccines in these four different multinational cohorts they have from these different countries, which is nice to see because they have different populations, different ethnicities in there too. This is another paper that came out a few days ago talking about again, boosters and talking about reduction in infections or severe diseases in children and adolescents. Is one of the plots. So basically, the conclusion that they have. This is the results they looked at. They looked at during the Delta period and Omicron period, and then they compared it to epidemiological data from patients, looking at different effectiveness, if they're infected, how bad they're infected, if they were admitted to the ICU, were they moderate to severe COVID, and so on. And basically, they find that indeed, within the 6-month period, like this year, Omicron variant declined 4 months after the first dose and then stabilized. And they saw things happening to these plots. But overall, they see that the vaccines have a nice, robust effect. As we, again, not a surprise to most people here. And this is a preprint that just came out. Came out a week ago. And this is a kind of a summary figure from Eric Toppel's Twitter page, but this is a figure from the paper. But this is something that maybe goes back to Hannah's question earlier. Is there long-term effects? There's other papers that show long-term effects for people who, let's say, had mild and they recovered and were fine. They think they're fine. This is from, I guess, a large collaborative group in the UK. I was looking at this. But basically, it's a preprint, so take it with a grain of salt. But in the paper, they're showing post-COVID cognitive deficiencies could be, they start noticing, for example, brain injury from mild to severe patients. They looked at 351 patients with COVID-19 who required hospitalization and then compared it to the normalization group. So I guess this is severe, sorry. There was another study I thought that was mild and severe. So this is severe COVID patients. So what they basically saw was that, this after... I forget how long after they were in the hospital. You have to read the paper, but the basic gist of it, they start seeing this gray matter volume reduction, injury markers, and things like that elevated in the blood. And the kind of the summary of this, we're equivalent to maximum 20 years of aging in these patients in the cognitive region from what they've shown. So they did a lot of imaging in the brain, looked at the different factors, and then they looked at some key markers. They didn't do like omics or anything, but for them, they had tau and these other markers here related to brain and seeing this type of issues, related immediate response. But that's kind of a scary thing, though, for people who are severe. Months afterwards, they see that there's a phenotype of 20 years of aging in the brain that could explain all the cognitive issues people have. Now, there are papers I've seen for mild cases too that have come out in the past couple of years that's shown similar, maybe not as severe as this, 20 years of aging, but they've shown similar kind of responses for people who've had mild cases of COVID. But this is a preprint, so take it with a grain of salt, but data is actually kind of impressive when they did this study. And they also quantified, what different types of neuro-COVID subgroups they had and what kind of phenotypes were developing afterwards.
Austin, I'll look at that paper later, but I do have a quick question. Did they compare to acute phase? So were there imaging, was there brain imaging done on those patients? And did they compare the brain images for long COVID to acute phase?
From what I understood, they only compared to like the normal controls.
Okay.
Yeah.
Because, there are substantial changes oftentimes reported in brain volume that would include both gray matter and white matter in acute phase infection as well.
Yeah, there were some papers that came out, a couple of years ago, looking at from UK. They looked at 6 months later after people didn't develop long COVID, but you're talking about same thing. Yeah, the changes occurred there, if I remember correctly. But yeah, here I think they only looked at as they say in abstract, for what I remember skimming through the paper, is that they compared to this number about 3,000 normal matched controls.
And do they give a breakdown on a region? So are there, you know, frontal cortex, hippocampus, brainstem?
Yeah,
etc.?
they talk about it in the page. That's what the image in the right here is. They talk about like the regions where they start seeing it. I forget the numbers, so we have to go in the paper and look at the figures. They talk about
Yeah,
that.
I'll do that later. Yeah, thanks.
Yeah. They have it in there, yeah, because they did the imaging, so they see the regions with exactly what you're talking about. What regions might have more of this reduction or the gray matter volume and so on, and the markers related, like inflammation and other things like that. If I forget, you have to go back in the paper. But yeah, I guess that's it for today.